Welcome, everybody. My name is Tommy Soares. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And today on Pathfinder, I have the pleasure of welcoming Dr. Jillian Wendel, who is the GLP Study Director at Labcorp, and that's in Greenfield, Indiana. Is that correct, Jillian?
That's correct.
Awesome. Well, welcome to the program. Thank you so much for agreeing to do this and letting us peek a little bit into your life and into your career trajectory. I got to tell you, I looked at your LinkedIn profile when I reached out to you and I said, oh, this looks like an interesting career trajectory. And I wonder about the interesting story behind it. So we're really glad to see you here on the program. I'm really glad that you accepted my invitation, and I can't wait to hear a little bit about how you got to be where you are today, and obviously where did you start, and your preparation to get you to be a GLP study director today in Labcorp. So take us back maybe just to the beginning, as far as you want to take us, maybe, and we can go from there. I'll ask you a few questions. How about that?
Sure. I ended up in science because I didn't have a choice. I was going to be in science in some way or another my entire life. I was a really interested kid that always asks why. And in fact, my husband hates the word why because I use it too much. But that's how I knew I was going to end up there. But I wasn't sure how. And in school, I was really good at math and science. So I actually thought the engineering route for a while. And I went to Rose-Hulman as my undergraduate program, and I started as a biomedical engineer and very quickly learned that I'm not as good at physics as I thought I was. So maybe I should kind of transition to something that I liked better. And I would say that's kind of the biggest story of my whole career path, is finding the things that I both liked and was good at, and then finding how to do that a little bit more next time and just keep moving along and saying, oh, that's the part I like. How do I have more of that in my life? So I transitioned from biomedical engineering to just biology. And with that, we were asked to do a capstone project. So that was kind of my first real dip into doing sciences and not just a school science fair. And I worked with horseshoe crabs. And they look like little absolute alien or prehistoric creatures. They're pretty ubiquitous on the East Coast, and we use them in the science industry to test for contaminants. So anything that's going to enter your body, whether it be a medical device or a compound, they need to make sure it's safe and doesn't have things that are going to hurt you. So we use horseshoe crab blood to test that. And at Rose-Hulman, we were trying to find a way to be able to culture their cells without having to just harvest it directly from the horseshoe crab. So I worked on a project with that for a while, and that got me really excited. And because of that, and my mentors, they encouraged me to go to graduate school. I started applying to both graduate schools and jobs, and I did get a role in graduate school. So I said, okay, this is what I'm doing. So I fell into that one a little bit, and I ended up at the University of Cincinnati and the Cincinnati Children's Hospital in Cincinnati, Ohio. So I didn't go that far. I started in Indiana, went to Ohio, and as you'll see, I came back to Indiana. But in University of Cincinnati, I was in a program that specialized in molecular and developmental biology, which basically means we studied everything, and you had to choose an internal specialty there. And I ended up in a neuro department and a neuroteratology lab. So we studied drugs of abuse and prenatal exposures and rodent models, genetic models for different diseases like overexposure to manganese, or I studied potential for having anxiety with a specific knockout model or predisposure to ADHD. So there are some genes that predispose you to that. And we did make a rat model that helps study that and, what options are. After finishing my PhD, I really enjoyed the rodent work. So I wanted to keep doing that. And I did again, look for both jobs and continued education, and was able to land a postdoc at IU School of Medicine with Shannon Hawkins, and that lab was part of the OBGYN department. So I got to keep working with rodents, and I got to keep working with animal models and animal development of transgenics, but I transitioned from neuroscience to gynecology, which might seem like a big switch, but I got to do the parts that I wanted. And in that lab, I was able to build on all of that in vivo or in vitro work by adding an in vivo component. So I had never done any of that before. And I got to do hands-on with cell culture. And that part wasn't my favorite. So I said, well, whatever I'm going to do next is going to have more of the animal side again. So I got to like keep learning and building. And I said, what parts of my postdoc am I really enjoying? I'm really enjoying the protocol design or experimental design, which is something that we still do in academia, but is also very active in industry and the CRO world. I enjoy seeing the project from start to finish, but I also liked not having just one compound or one gene of interest that my whole life was focusing on. And moving to a CRO helped me be able to do that, because now I get to have lots of different things coming at me and lots of different people who's like, this is my baby. And I get to say, okay, I'm excited to work on your baby for a while with you too. And we'll do something really cool with it. And then I'll give it back to
you. That's awesome. That's so great. And seriously, what an adventure it seems so far, because it's so varied and it has so much variety that I'm fascinated by just the way that you've been able to move through flawlessly. And what I really like is that you've seen at every step of the way things that you've enjoyed, and you've gone after more of those things. And I hope, I wish people that are watching this program can do a little bit more of that. If there's something that you're good at and you like, because you're good at it, or you have an affinity to it, and you end up being good at it, it's a great way to find a lot of pleasure, right? And some of the work that we do. Tell us though, you said, oh, I realized I wasn't that good at physics, so the biomedical engineering route it wasn't. So, like, was that a particular moment where you're like, oh, after an exam, or was it a time where you had doing a project and you're like, oh my gosh, this really isn't for me? Like, can you take us back to the realization that, this is not the way to go? At least not for you, it
Yeah.
wasn't, right?
Oh, there's a class. I remember that class. It was called Statics of Mechanics and Materials, and we had to draw free body diagrams in every single homework, and I could not get the arrows right ever. I remember having a whiteboard because I couldn't do my homework on a piece of paper because it was not right. So I would do it on a whiteboard and I would draw everything out, and I always had a homework buddy, usually someone that was higher up in classes already, that would like work with me because they wanted me to succeed too. And I'd be like, okay, this is what I got. And they'd go, you don't even have the same amount of arrows pushing up and down. And I was like, there's a God force. I don't know. So that was definitely the class that I went, if I am putting in this much effort and I'm struggling this hard, instead of continuing to struggle, what part of this can I enjoy and take somewhere else? Because I did really understand and really like some of my other physics classes. So it was clearly like forces were the issue. Fluids was fine. I liked fluids. So I went more down that route and said, okay, what's a little bit more like this? Chemistry. Let's do more chemistry.
That's great. And I think it happens to all of us, really. We come to a fork on the road and we say, well, it doesn't seem like this was the route that I was intended to take anyway, so I'm going to keep going in this other direction.
But there's a difference between being hard and being too hard. And that's why I did make sure that I had those resources so I could try to get through it. And I did. I passed the class. I didn't fail it, but I did say that wasn't worth passing it.
Yeah, and again, I also think, you hear the recommendation that if it's difficult, then go work on that, because you'll get it at some point in time and it won't be as difficult anymore. But as you're saying, like there is difficult, and then there is like, well, interest and challenge need to come together as opportunity, right? Um, so
Absolutely.
there is that balance. I'm sure you've done difficult things too throughout your career that you really had to put it in, and really hone in or feel like a novice again. I've heard that this is a good thing for us to challenge ourselves, to feel like, oh boy, I really have no idea what's going on here. Hindsight, what would you have done differently, knowing now what you didn't know then, about how to approach that if you wanted to succeed in that in that class? What do you think you would have done differently?
I think knowing what I know now, I would have just tried to go into it more open-minded. And that's a reoccurring lesson that I've had throughout most of my careers. When I go into something and I either think I'm going to be good at it or I'm not, it creates a hurdle. And I have to get past that hurdle. Or if I go into something and say, I'm not going to be good at this, I'm not going to enjoy it, I'm not going to like it, then I also give myself a hurdle. And then I'm battling myself. I'm like, well, I could be good at this, but I'm already angry about it, or I'm already really grumpy about this. So having a different mindset and trying to go into some of these situations, either if I don't have a lot of information, saying, okay, I'm just going to see how it goes. And if I do, try to learn what I think the problems are going to be and decide if I think that those are okay before I even start something new. So every field you go into or every space you go into is going to have problems. And it's which problems are you okay with versus which problems are you not. And that's how you know you're in the right field, or you're even in the right niche of that field. Is, oh, those problems are fine, I can tolerate those. Too fast timelines, I'm great with timelines.
Yeah. And having that confidence in yourself, right? Of knowing that no matter what challenges come your way, you can handle. You spoke also about your mentors at that time, and maybe even mentors as you went into grad school. Do you remember some of the lessons that they gave you as you were moving through that time of, well, what should I do? How should I proceed? What schools, and so on and so forth, and where should I go?
So I have had a lot of really good mentors. I've been very blessed in that sense. And I've had a lot of very good mentors that were my assigned mentor, but I also reached out to people that I felt had similar interests and used them as a mentor, whether they knew it or not. But I was like, I'm watching you, I'm learning from you. So having both of those assigned and just kind of freestyle mentors has been really important. And I've gotten very different things from them. Typically, my assigned mentors will encourage me to do things on my own. So I'll come to them and their response will always be, well, what do you want to do next? Or what's your first step? Instead of, telling me exactly what it is. And some of my ad hoc mentors that weren't assigned really like to remind me, don't do it. And that's a phrase that I need to hear all the time because I'll get myself into too many things. I get excited and I get too deep or in the hole. So having people say, well, what do you want to do next? And also, maybe don't do this extra thing right now has been really helpful in my career.
That is so important, isn't it? At first, the recognition that we're excitable, that we just get, and if you're anything like me, I just get excited about the different science and the different things that are being discovered or being done, and the different opportunities and people that are doing these things. So it's difficult to say no. How have you learned to say don't do it, or uh, don't do this one now, go after that one. How have you learned that? Because it's hard. It's difficult to
It
say
is. It's really easy to get excited. So it's about taking a step back when somebody reminds me, don't do it. Essentially, what that says to me is, do you want to do it? Does somebody asking you to do it because they can't, or they don't want to, or they're delegating, or why is this coming on your plate? And if it's something that I really personally and passionately want to do, then I can still do it. I'm still absolutely keep time and space open for my little passion projects that might not be part of my job description. And I keep space open for when maybe a manager comes to me and says, we really need this work done, and we really think that your expertise can help lead this project. And those are reasons to absolutely say, okay, I am going to do this. And I thought about the don't do it, and I chose that this is still, I have the time or the mental capacity and the joy for it. And other times I see, okay, well, I'm potentially doing it just because I don't want it to come back later, or I'm afraid somebody else will do it and I want the credit, or any reason that might not be a great reason. And it just makes me challenge that and say, why? So I guess it goes back to why, but it's phrased a little differently.
It's so important though. And it's something that I don't think we hear very often, Jillian, in that, asking that why, stepping back and asking that why. Um, Many people are asking themselves why not instead of why so. But I think that's very important, that the way that you phrase it, because it allows people permission to say no, right?
Mhm.
And giving yourself that permission seems to be many times a, a thought of like, this is how much time I have, and I don't have any more. People say, "Oh, I'll make up time later," but I'm sorry, I haven't seen anybody who can make time. We
Mhm.
all have the same 24 hours a day, so it's so important, so important. Thank you for those tidbits, because I think many of us need to hear those words. Moving on to this neuro-based project in Cincinnati at the Children's Hospital, working with rodents and models and like all of this, like it must have been a different world. It must have been probably scary too, because it's like a lot of brand new stuff, new people to work with. Take us back to when you moved to Cincinnati to start that work, and like, how was that?
Well, I saw my apartment for the first time the day I moved in, because I just kind of picked something online and moved in. So it was
Uh-huh.
all a little scary as a whole, but my program did a really good job of having us do rotations. And I think most graduate schools do now, of where you take your early basic classes and you start working into that curriculum. And at the same time, you're rotating through different labs and trying to decide. Well, during one of our classes, they also made us rotate through every animal model as like part of the lecture, as a hands-on part of the lecture, so we could experience different animal models. So I was able to cross off a chunk of potential labs, because I was like, I don't want to work with this animal model, or I don't really want to do this one. Oh, I liked this one. Who has labs like this? So that really helped. And then doing those rotations and having those rotations be a forced number, as they said, you have to do at least 2, and we want you to do less than 5, please, because otherwise you'll never choose anything. But having to like do more than one, and they encouraged 3, was to make you try a couple different things and not just pick and settle with the first one. And I think that was a really important thing, because you learn something in each lab, and you learn people and different styles in each lab, and you start to learn, well, what style teacher is going to work best for what my needs are. I need somebody that's going to push me, or I need somebody that's going to have pull me back, or they're going to let me run free and then help save me when I ran too free. So learning that is important, and you can get that through rotations. And it made actually choosing a lab less scary, and choosing a lab both based on the people, but also on the type of research. Because I knew when I joined, most of the people that were in the lab weren't going to be there most of the time. They were a couple of years into graduate school already. They were going to be transitioning out. We were going to bring new people in. So I didn't want to choose a lab specifically based on those, I guess, temporary positions that people were in. I said, what type of research has this lab published with repeatedly? And is that something that I want my name assigned to? And that was a time in which my mentor was transitioning from having just a lab to also being part of a core facility. And him and one of his colleagues started the development of the behavioral animal core. So they were not only going to be doing their own private research, but they were also going to be taking on other projects that were less informal and definitely way more formal after it became the core. And I was like, was this something I'm interested in? Because this means I might have a little bit more responsibility outside of just my work, but I would get so much exposure to people that aren't just working on neuroscience, but might need that portion, or might be working on something muscular and need to see if there's a behavioral change there. So that was where I started to like tiptoe out of my little neuro world by being able to see it in this safe space that was the core facility too.
And so did you get an inkling, or did you get to also see some of the administration of the core facility and the business part of things, and charging, and how much should we charge, and the rates and per diems and all of that stuff? Did you participate in that process as well?
I did get to participate in some of that, especially as it was getting started. It was part of our general lab meetings, and even the scheduling of how do we prioritize different aspects, and how do we make sure that everyone that has this behavior for the core facility gets completed, but we're not getting in the way of somebody's dissertation work that they started years ago. So that was definitely some learning curves for everybody on how to integrate these two together seamlessly. And because of being part of this, I got to interact with vendors, and that was an experience that I hadn't really had, or might not have, of bringing in new behavioral tasks. So more than once we would choose a room that was in the facility and completely change what behavior was being run in that room. Get something in from a company that maybe they had a published paper, but we had to validate it in our space, or write an SOP so that we can be really consistent on other people's studies there. And that was something that was helpful, that I didn't know was going to be so helpful for my world now, but was helpful in my postdoc also, because we talk about SOPs, or standard operating procedures, a lot, but sometimes we wander away from them a little in academia, whereas in industry and the CRO, like they rule your world. So it was nice to like dabble in that and get an idea of what that means and what those look like and how important they are.
Did that exposure, you think, make you more competitive for this industry role that you're playing now?
Oh yeah.
So it helped
I
you,
do.
it helped you get the job that you have potentially. Yeah.
I absolutely believe that. So
Yeah.
both the personal connections and people knowing who my mentor was, his name is Dr. Chip Voorhees, and him running that behavioral core, but in being involved in neuroscience, that helped. But also the experiences I had from it, and the diversity in the types of experiments, being able to come in and talk about experimental designs that I've had, or because of the core facility, we even ran some things following some of the international guidelines. And I was able to talk on that, because they'd say, well, have you run GLP studies? And I was able to say, I got GLP trained because we had people come in and say that they needed this. So I know what you're talking about.
Yeah.
And that helped significantly.
For sure, for sure. It's so interesting that you're mentioning this experience, because the people that are watching, they probably don't know, but our next few shows are going to be interviewing Pathfinders from core facilities, because I did want people to understand that that is also an option. It's
Mhm.
available here in Indiana. And not only is that an option as a career, but as you said, if there is an opportunity for you to experience or pick up a technique or so from a core facility, it's not a bad thing. It's always good to know, and to know the operations, and right. I mean, just
Mhm.
and I'm sure, like, you probably didn't operate in GLP capacity, but you knew what you needed to do in order to be
Mhm.
compliant with GLP regulations. So tell us a little bit about, like, and I'm sure you are so proficient in it now, but for people that are watching that don't know this good lab practices term, and what does it really mean, and what does it entail, what
Mhm.
do you say was your exposure then that gave you good runway for what you're doing now and the SOPs that you're following now? Give us a little bit of that, Jillian.
Sure. When I first heard about it, I don't think I had a whole lot of background knowledge of the why behind it. It would just seem like this extra set of rules that I was like, aren't we kind of doing all of these things anyways? And honestly, most labs are doing most of those things anyways, because if it's written into the good laboratory practices, it probably means your experiment will be better if you do it. So that was a plus when you finally start digging in and you're like, oh, I'm doing most of this. But
Yeah.
the entire intention is at one point, it was decided that there was a little too much flexibility and freedom in how we were holding science as a standard, especially when that science was moving from non-clinical into clinical, and saying, do we always trust this? Do we always trust the facility? Do we always trust the work? How do we have some consistency across? So the good laboratory practices were developed to create some of that, to say this is our standard. And if you're not within our standard, you need to tell us you're not within our standard, and you need to tell us why and if it matters. So sometimes we don't run things completely based on good laboratory practices. And that just means we have to say it. We have to write in our reports, this part didn't follow it, and this is why. And those good laboratory practices, it's a little booklet, and it's not too long or hard to read, but it outlines what people are important in the facility, what documents you have to have available. So every study needs to have a protocol, and that protocol needs to be reviewed by certain people, and it will outline that, and it tells you what it needs to be in the protocol, so that every study you can go back and you can redo it. And that's this importance of all of this. If you can't reconstruct your study based on your protocol, then you're missing information. So that's what they're trying to get at, is saying we need to be able to reconstruct this. So have your protocol, have your people review it, have your approvals on it. And then it also talks about the reporting side, and it says, whether or not you followed everything in your protocol, we need to know that, because sometimes you don't. Deviations happen, and sometimes deviations are a good thing and sometimes they're a bad thing. But noting that you deviated from that protocol is important, or that you made an amendment to that protocol and why, and being able to put that in your report, because your report is going to tell you what your answers were, your protocol is going to tell you what you did. So it outlines both of those documents, and it also outlines, other basic standards of don't do anything unless it's either written in the protocol or written in a standard operating procedure or a policy. So you can have both. And those are saying that this is a task that happens so frequently within your facility that writing it out in your protocol every time would make your eyes bleed. So instead, we're going to let you have a separate document that's going to be maintained in facility records. And you just need to be able to have that, prove you have it, and prove that you followed that SOP. And with that, like, body of documents is how you be able to have that reconstructability, that no matter what agency you're submitting to says, oh, you followed these. Because although the good laboratory practices that we follow are with the FDA, the Federal Drug Administration, or the EPA, Environmental Protection Act, they also are similar to the GLP standards in other countries. And we actually have an agreement with other countries that say, if we follow our GLP standards, are they good enough for you? And they say yes. We've also reviewed yours. Ours look very similar too. If you're following those, we'll accept your studies too.
that standardization must be, it's such a gold nugget, really, to continue to polish, right? Is there ever any changes? Do you ever get any FDA or any other body entity saying, okay, this is how we used to do this. Now we are adapting it this way because, I don't know, now we have computers. And before, when we did it, we didn't. That type of thing. Are things shifting in the GLP world, or are things fairly standard and they remain the same and not really moving at all?
So there's a couple of levels to it. That first good laboratory practice, that hasn't changed in quite some time. And we did add an addendum for electronics and how to trace that electronics and how to make sure that you have control change and you can see when something changed and why, and have an audit trail so you can never delete anything. And there was a lot of information about that and how to have signatures that aren't on paper. So there was an addendum for some of this technology that was being added. But after the GLPs, you typically have guidelines, and guidelines are going to be based on who you're submitting to and what they want and their requirements. And those will change a lot more frequently. Well, every 10 years, maybe. So they're still not that frequent, but they will come from, a smaller body of researchers getting together and saying what is changing in the world around us, what technology is changing, what assays are changing, and what do we need to update. So we've had some recent updates to how we run carcinogenicity studies. So recommendations in those guidelines. And we've had some changes into how bioanalytical methods are developed and what we need to put in bioanalytical reports to show that we have all of the controls in place. So that's the level that you see a lot more change as opposed to those GLP standards level. And we change SOPs all the time.
Yeah. So, and are you able to then, in your role now, are you able to be a voice for change? Are you able to be able to say, hey, we've observed this and we should really change that guideline to reflect this better? Are you able to have that voice?
So I personally am not on any committees that are participant in that, but I know a lot of people in and out of Labcorp that are, and they do like to have voice from CROs and industry and academia to hear what everything is. And it is a committee of peers. I have been involved kind of on the side in some conversations by being part of different memberships, so different like programs or different conferences, and going to some of those and meeting with people and discussing what the problem is. And it turns out that like the person leading that talk is on the committee that's working on this new guidance, and they're sitting there like listening to all of us be like, this is a problem or this doesn't matter anymore. And they're the ones that go back and take it because they show up at conferences too and like to overhear it all and know what's going on. So I'm involved in more of that side of things.
Yeah, that's so cool. That's awesome. And just to be able to hear you talk about the way that you interact at conferences is also great, and probably eye opener for some people too. I I don't want to fast track through the IU experience. I do know that there was probably some really good stuff there. And you mentioned cancer. I have to bring up your endometriosis and ovarian cancer and so on. Tell us a little bit about that time in your life, and because for me, postdoc and that period of time is such a special moment where you're not a student, you're much more independent. It's really up to you and the science that you do and what you pursue. So take us back just a little bit to that time if you can.
Sure. I would say being a graduate student is all about being all of it. You know every bit of everything you're doing. You are the one and only person doing any of it. And postdoc, you're really transitioning into not being the only person involved in your stuff, and learning how to have those collaborations and just say, I need to know this because it is part of my project and is really imperative, but this is a skill that would take way too long or too much effort for me to become as an expert as you are at it. Can I partner with you? And learning how to have those conversations and how to balance, both people's time and their interests and their commitments, but being able to get that extra information onto your study to be able to share it as a much bigger picture was a big change between graduate school and postdoc for me. But it allowed you to then kind of test the waters in a lot of other spaces too. So I came in and there was a project available to work on an animal model of spontaneous development of endometriosis. And I was like, yes, this sounds so cool. And it was really cool. So it's a mouse model. And when we were working on it, we said, well, we really need to understand the mechanism behind this. And that's where some of those collaborations came in, because there started to become this immune side of it that I was like, well, this is kind of cool, but go down immunity and all of a sudden there's these massive pathways that I don't think I quite understand. So worked with a graduate student in another lab, and she ended up being part of that research by aiding her expertise in it and building that relationship and reaching out to core facilities. So again, the core facilities. So because of this project, it actually branched off and went towards in vitro. And I started working on 3D modeling. Or 3D cell culture. So we would take cells that you normally grow on the bottom of a plate in a single layer. And if you instead put them on a round bottom plate that doesn't have anything for them to adhere to, they stick to each other and they form these nice little spheroids, is what we call them. And they're just little balls. And there's a core facility at the IU campus that will take those balls and they pick them up with a vacuum and stick them on to these needle skewers. So they're metal needles, and it's called a kanzan. It's based off of the Japanese floral design, and it's basically shish kebabbing your cell culture spheroids. But because they are then kind of near each other, they fuse together like the spheroids did, and you get this bigger construct. Because a computer is printing it, you can do it in whatever pattern you want. And then after a couple of days, you can pull that whole construct off of that kanzan array, and then those holes will fuse too. So now you have this completely scaffold-free 3D cell culture, which was a really cool experience to work with the lab on. And putting that in perspective with the mouse work kind of gave this really intertwining of mechanism and also in vivo functionality and challenging that model and being able to use some parts that I was really good at and build some parts that I then became really good at, and then some parts that I am versed in, but I'm not an expert at, and being able to say, again, which things are worth becoming an expert and which things are worth utilizing the resources around me and utilizing them appropriately so that I still have contacts later.
Yeah, absolutely. So interesting, though. I love the 3D cell culture approach, and I really like that you got to participate in it. It's so rare that we stop and realize how many important accomplishments we've had until we go back and reflect on these times where we participated in technologies. And we forget how few scientists there really are in this world right now. I know it seems like there's a lot, but I heard a statistic where it says that 90% of all the scientists that have ever existed in the story of humanity are alive today. And that tells you that there is a really small community. We're just getting started here. And to be able to participate and be a part of that development must have been thrilling. I hope you're as thrilled about it as I am right now. And looking back at, wow, you're like a pioneer in 3D cell culture for doing that type of work.
It was
Uh,
a really cool space to be in. And I don't even like in vitro, but it was still a really cool space to play in for a couple of years.
Awesome, awesome. And then like, I don't know if you combined it with like laser capture so that you could get out one cell and start characterizing it out of the bunch and looking at how the periphery cells were doing versus the cortical cells. And like, anywho, it's so neat, so neat. No wonder you ended up where you are. But tell us a little bit about, how did you approach Labcorp, or were you approached by Labcorp to be in this position, or to get a job there?
I approached them. So it was reaching out through just a job application on their site. I knew a number of people that had previously worked there or were still working there. So I did have a little bit of an idea of kind of what the space was. But the Greenfield site, so Labcorp is a global company. So it's also why we're kind of well versed in some of these other countries' expectations and things also, is because we have sites all around the world. And our main site in the United States is in Madison, but we do have one in Greenfield, Indiana, and in Somerset that do the non-clinical research. So at the time that I was looking to make a transition, I found out that this site was trying to move towards having a business line of crop protection and chemical and not just work on pharmaceuticals. So there was this new space opening in this new niche that I could learn all of the pharmaceutical work, but there was crop protection and chemicals, which kind of aligned in some ways with what I had previously done in grad school of early exposure to environmentals. So I said, oh, well, this kind of leads me back even a little to some of the stuff that I was doing in the past, which I did very much enjoy. And maybe I don't have to give that up. I just put it away for a little while and can come back to it. So that's where I got interested in coming into that space. And I did look at a couple of different roles at Labcorp, and the study director role seemed the most appropriate for me at that time because of the fact that you get to wear a lot of hats. And that has always been something that I've enjoyed. And that's something that's very true in academia also, especially during all of your training, as you wear all the hats, or you're learning all of the hats. You wear the baby version of all the hats. But that was part that I liked. So taking something that I liked in one part of my career and encouraging to continue to do that later is why study director sounded interesting to me. It was giving up lab time, giving up that hands-on. And that was probably one of the biggest kind of internal debates I had, is so much of what I enjoyed of being in academia was the hands-on portion. I loved pipetting. I loved doing mouse surgeries. I loved all of that. It was cathartic, or doing a necropsy on a mouse. It's like, well, I know how to do this. Or a bad day and I'll put my headphones in and I'll just go to town PCRing for quite some time. So it was kind of like, am I willing to give this up? But it was the challenge of, can I build really good experiments that somebody else can read this and then do it the same way that I would, so I don't have to tell them, oh, just let me do it myself. So that was how I was challenging myself in this new way, is, okay, can I write it good enough?
Incredible. And was it difficult then to let somebody else do it, and then you be like, what are they doing?
Yep.
Did you want to go into the lab and peek to see how it was going the first few
Oh, I
times?
go in all the time. I just am not allowed to touch anything.
Okay, so now we know that the GLP study director is really not allowed to touch any of the equipment. Okay, and so, but it must still be so enjoyable. What are some of the things that you love about your job now? And maybe I do want to say, okay, so I want you to tell us a little bit about the interview process if you can, because I know there'll be people interested in applying to jobs like this, and just knowing about so tell us about the interview, and then tell us a little bit about what you like doing in your day to day.
Sure. So most, large companies follow that STAR, the situation, task. I don't remember what the other two letters are for, but it's a story around the question. So they'll challenge you that way. And Labcorp was not any different. The way my interviews worked is it was during COVID, so it was all virtual, but it was a number of panels or individuals. So I met with all of the management within my department, and I met with a number of the senior study directors. I also met with some of the people that I would be working with, so not in my position, but senior study coordinators, to say, oh, we could work with her, or we would have to work with her every day and that would not go well. And I met with some of the people in operations. So everybody kind of got a say in it, or at least got to like be part of the conversation. And it was all very structured in the sense of they would ask you about a time in which you've experienced something or had a challenge. And it was about not just you telling the story of it, but what you learned from it, or what you would do differently, or I've never experienced that, but this is how I would start it. This is how I would go about being part of this discussion or moving forward with it. And I would say in a lot of ways it was kind of similar to like maybe your qualification exam, where they just ask you questions until you say no, or I don't know, and then they ask you, well, how would you try? It's similar. And that, but so much of it was about learning kind of the culture of Labcorp. And for me, so much of my questions back to them were about different leadership styles and what is the team dynamic like, and what are some of the expectations. So I guess I challenged all of the managers that were on there and asked them, what's your management style? And I think, joining later, they were like, yeah, that was interesting. But it was important to me to make sure that that was going to be a good space, and having the management style that I needed was going to be important. So they knew that I was actually very serious in considering it. And they also got to learn how I interact with people when I need something or I want something. I can be a little pushy of, give me that answer.
But I think you raise a very important point again, and thank you for doing that, which is, if you're going into an interview, make sure you have some questions ready for the people who are interviewing you, because you think it's all about you answering questions, but many times they're also judging you on the types of questions that you're asking them. So, I mean, I'm sure it was a challenging question to ask on the spot. What is your management style? I don't know. I wake up in the morning and I go to work. But I think that is so important. Were there scientific questions that you also asked, or were they mainly operational, administrative questions? What are some of the other questions, if you can remember, or if you can share?
So there were some like study design questions. So them wanting to understand you did a PhD, but did you really, design your own experiments, or did you have a lot of help with it? So kind of probing in deep enough. I remember my manager at the time, he specifically said, I don't want to hear about these first two projects on your resume. Pick a different one. And I was like, wait, what? Or, he asked another follow-up question or a new situation. He said, and it can't be from one of the projects you've already talked about, with something that you learned from another one. And I was like, ooh, I got to dig kind of deep in here. So just being really prepared. And I mean, it's your life, it's your experience, it's your projects, everything about them. But most of the time when we do those like practices with the people around us to get ahead of time, you talk about like the big one that you work on. Well, sometimes they're going to say, no, you get to talk about a secondary one. Let's see. Okay.
Yeah, that is wicked on their behalf, but it does show, and it goes to show that being prepared is a big battle here of the interview process, right? I say this because it's a time before you go in there, is a time to reflect on what you've done, what you've learned. And even if you don't spell it all out, there are certain things that you're carrying with you that you've learned through that process, right? And you have to recall those on the spot, right? I guess they
Mhm.
want you to, they want to see how you're thinking on your feet, right? Was it
And that's is so much of what it is. It's not just about your experiences and are you technically ready for this, but those questions are designed to get at how you handle situations so that they can decide if the rest of the group you will fit in with. Because we do work on our own projects very individually, but we use each other as resources constantly. So making sure that the team fit is going to also be appropriate, and how you're going to work well with the operations groups, and the fact that we are ownership over the studies, but we have no ownership over any of the other teams, but we need to be able to communicate to them our needs at all times, and what this project needs. So can you communicate that in an effective way that is also teaching or graceful? If you're asking them to do something very different than they're useful, being able to explain why. Back to that why. We really like why.
absolutely, and the communication piece so important and keeps being raised here by pathfinders that are on the show, that keep saying, it's so important to be able to communicate and communicate to different audiences, different people that understand, or they understand different pieces or different parts. You made a point, Jillian, of saying that in your time in academia, you had this opportunity to rotate through the different labs that were specializing in the different models, and that was awesome because it really let you see and let you identify with what you would be attracted to working on. Was there that opportunity at Labcorp, or was this job, um, that was it, and there was really no, there, you didn't really get to rotate, or was there a rotation like part of the orientation as you started to become part of the LabCorp family?
So I would say there wasn't a rotation through different roles, but there's definitely exposure to different types of projects and learning models that you kind of fall in line with the best, or compounds or types of disease models. That's what I get to rotate through. And that's what I made a big deal with my current manager of saying, hey, I want to try them all. I really want to just put my toes in the shallow end and then figure out which ones I was good at and which ones I liked. And then we'll try to find a specialty after that. So I work in pharmaceuticals, and I work in crop, and I work in chemicals. So I run studies for all of those, and I run studies that are considered too early to fall under GLP, and I run studies that are GLP, and I run studies that are really late, so it's already in clinical trials, and we're doing follow-ups, or we're doing those long-term carcinogenicity. So I'm doing kind of the whole thing, where there are some people that say, well, I want to find a specialty right away. Oh, I love doing this intrathecal dosing. I want to learn all about this. This is a specialty. Or I want to work with a client and I want to be their alliance SD so that we build a really good partnership and we have our own lingo. And so you can go kind of any way you want. You can learn a specialty, or you can have a little bit of all of it. We're still
I
figuring out what I'm doing.
well, but I love that you have this variety of different projects. It must keep you so active and challenged and just amused with doing the work, because yeah, there's no way you can get bored doing this work that you're doing.
Definitely not. And when I get a new project in, we have, a kickoff meeting with that sponsor. And I always like to tell them, get me excited about your compound. And oh my God, do they get excited to tell you about it? And they're like, oh, okay. And they go off and they're like, this is what we've been doing. This is how it's working. This is the mechanism behind it. This is what we want to do. And I'm like, okay, let me type faster. And then I can take that and I can take that all and like get it some into their protocol so that we're making sure that even if they didn't ask for something in the beginning, hearing that conversation, be like, are you sure you don't want to add this assay? Or pass that information down to operations and be able to put something in the animal room to say, this is why you're doing this. So that anybody that walks in is like, oh, this is really cool. And they want to come back and ask questions, or they say, hey, will you host an ad hoc meeting after the study is over and tell us what happened? So we'll do stuff like that because of how excited people get us about their compounds or the model they're using or the different study design they're using. So no, I'm not bored ever.
That's great. And Jillian, tell us, are you only, you mentioned that you're a global corporation, and I've admired Labcorp for a long time. It is an awesome company, and the whole Covance, LabCorp unification and all of that. I mean, I've been here since that time, and I've seen that whole thing come together, and it's just been awesome. Do you get to work with international clients too? Do you get to run studies with international? So does that mean that like you also have to now learn regulatory practices in the different countries that are supposed to be you're delivering the data for? Is that how it works? Tell us a little bit about that.
So if we're running it here in the United States, we're compliant to the United States, so the EPA or the FDA. But if we know a sponsor is intending to submit their compound outside of the United States, we're compliant to the United States, but we will add guidelines from other countries also. So sometimes you have to take all of those guidelines and try to balance them out. And a lot of the times they're very similar. In fact, a lot of them are word for word the same because they have met with each other so many times and said, hey, we like how you did it. Let's write that into our guidelines and have that global harmonization. But knowing where your sponsor wants to submit their work and when and who they want to submit it to first can sometimes be a challenge because different countries might have slightly different expectations or be a little bit more difficult with this type of compound to submit or want more information. Or what we run into is different countries have different expectations with how we do research in animals and being very sensitive to that to say, this is something, this number of animals would be inappropriate to submit to that regulator. So let's make sure we're reducing the number of animals or increasing it because they want higher power to prove that it is safe before they go into humans. So knowing those kind of nuances more so than being compliant to them, because we want to make sure we give the client exactly what they need so they don't have to come back and ask for something else later. Unless they're giving us another study, and then we'll totally let that happen.
Yeah, but what I mean, especially because you deal with ag products too, ag chemicals as well. I mean, I know the European restrictions are much higher than the American restrictions, which are much higher than, let's say, South American restrictions. And so you're dealing with a lot of these, differences in standards that I think, would create the challenge. But what I love is that you just take it on as it comes, and yep, no problem. You have this confidence and the variety in the different projects, the variety in the therapeutics and the teratogenic compounds, the variety in the ag chemicals, and like, my gosh, it is so awesome to see you thrive in this way and be so excited about the work that you're doing and sharing a little bit with us. If people wanted to find out some of the opportunities at LabCorp, what's the best way to do that? And could they link in with you on LinkedIn and maybe ask you a few questions there? I just think again that you've provided a window of what could be in somebody's career, especially if they have all of these tools and variety of different molecular biology or preclinical trials or whatever it might be that it would be exciting for them to apply. So if you can, just what's the best way to do this?
So if you're looking for roles at Labcorp, it's labcorp.com/careers is the best place to show you, even the ones international. It's going to show you the whole gamut of what's available, and anyone is absolutely welcome to reach out to me. LinkedIn is a great place. I think it took me like 24 hours to respond to you, so I'm a little active on that, sometimes more than I should be, but I keep up with it. I keep up with people, and I have worked with people still at IU to have them consider transitioning over to Labcorp or collaborating with them or checking in. So there's definitely still a lot of interaction.
Awesome. Well, Dr. Jillian Wendel, thank you so much for being our Pathfinder today, GLP Study Director at Labcorp in beautiful Greenfield, Indiana. Thank you so much for all your guidance and wonderful advice today.
Thank you so much for asking me. It was wonderful to be here.
Yeah, have a great day, and hopefully we'll catch up again soon.
Thank you. Bye.
Bye-bye.